faculty of their department, namely Dr. Sai Harsha, Assistant Professor, Department of Pharmacy Practice, JSS College of Pharmacy, Mysore. And with the help of Harsha sir, we got today's speaker, and we are happy to share this moment with our institution students and faculty. So now I'm requesting Vijay Institute of Pharmaceutical Sciences for Women, Department of Pharmacy Practice faculty, namely Dr. Tabitha Sharun ma'am, to introduce today's speaker, Dr. Jihad Syed. And this is a good opportunity for our student to understand how the pharmacy practice qualified or PharmD graduates are having a bright future to do so many jobs, like whether after completion of PharmD you can go for a research or a pharmacovigilance job or teaching, etc. So with these few words, I am requesting Tabitha Sharma ma'am to introduce the today's speaker to the students and other faculty members of our institution. Thank you. Sir, yes sir, yes you are, sir.
Thank you, sir. Good morning, one and all. Myself, Dr. Tabitha Sharon, Assistant Professor, Department of Pharmacy Practice. I am so pleased to be with you this morning and to have the distinct pleasure of welcoming our distinguished guest, Dr. Jihad Syed, to share her insights and experiences with us. Our guest holds a BPharm and PharmD PB degree from JSS College, Mysuru. Currently, she is working as an Assistant Professor and Senior Research Fellow, Department of Pharmacy Practice, JSS College of Pharmacy, Mysore, Karnataka. A testament to her dedication to the pharmaceutical field and her commitment to improving healthcare. As a PharmD graduate, she has undergone rigorous training in pharmaceutical sciences, patient care, and medication management. Her journey in the world of pharmacy has been marked by academic excellence, clinical expertise, and a passion for making a positive impact on the lives of patients. Because of her hard work and dedication, she had a meteoric rise in her career from working as a pharmacist at Bharat COVID Suraksha project to a clinical research coordinator, where she involved in various phases of clinical trials like MR vaccine, COVID-19, ICMR COVAX. Her academic achievements include Best Student Award from Association of Pharmacy Professionals, Student for Best Evidence Pioneer from the Cochrane, UK. She also received travel grants from DST and served to attend a conference held in Denmark. She has received a scholarship from Tata Trust Medical and Healthcare. Her journey serves as an inspiration to all of us, especially to those who aspire to make a difference in the field of pharmacy. She has likely faced challenges, embraced opportunities, and gained invaluable knowledge along the way. Today, we have the privilege of learning from her experiences, gaining insights into ever-evolving landscape of pharmacy, and understanding the crucial role pharmacists play in healthcare. Without further delay, let us extend a warm welcome to our esteemed PharmD graduate who will undoubtedly inspire us with her words of wisdom and her journey in the world of pharmacy. Dr. Jihad, thank you for being here with us today.
Thank you, ma'am. Thank you for the warm welcome.
Now we are handovering the section to today's invited speaker, Jihad Syed ma'am. Ma'am, please start your lecture, ma'am.
Oh, okay, sir. Thank you, sir. Is the screen visible, sir?
Yes, ma'am, it is visible.
Okay. Okay. Good morning. Thank you for the warm welcome and thank you for this opportunity. I would like to thank even my supervisor, Dr. Sri Harsha sir, for his guidance and his, mentoring that I am here today delivering a talk in front of you. So today we'll be talking about confirming signals in pharmacovigilance and the job perspectives in pharmacovigilance. So as now we are having all the PharmD grads students in front of us, this would be a very helpful topic for all the 5th PharmD students especially, because they have the pharmacoepidemiology subject in their syllabus. So first, as a healthcare professional, we have one concern, that's usually the risk benefit of a drug. So are the drugs safe? So we will see that a drug undergoes rigorous, monitoring and regulated, safety evaluations during their randomized control trials. That is, the molecules come from the preclinical, then to clinical, in the clinical phase 1, then they go to the phase 2, and then the phase 3, and then from the phase 3 into the market. So in these things, usually there are few limitations. There are limited number of patients. So they are going to recruit certain eligibility criteria, which they are going to pick the patients that they have to adhere to eligibility, inclusion and exclusion criteria. And they have a specific study duration. So after this, the drugs are introduced into the market. But however, these results from the RCTs do not represent the real world population. Because in the real world, when the drug is into the market, everyone are going to get your drug. Like it's going to be an old patient, it can be a pregnant woman, it can be pediatrics. So all the special populations and everyone are going to receive it without any, strict eligibility criteria. So now we don't know how the drug is going to react into the market when there is a wide variety of genetic pool that we are sending it in. So now what are we going to do? So how are we going to monitor the safety and effectiveness of a drug after marketing? So that's something called post-marketing surveillance studies. So in this, we have hypothesis generation or hypothesis confirmation. So hypothesis is something where we are assuming, like we are drawing this one, that this drug may cause a problem. So we are assuming that it is going to cause a problem. And now we are going to follow it up to see if really it is going to cause a problem or it's not. So regularly, we are going to have two subjects that are going to go hand in hand. That's pharmacovigilance and pharmacoepidemiology. So as we know, pharmacovigilance is a science and activity related to the detection, assessment, understanding, and prevention of the adverse events or the adverse effects of any drug or vaccine. Here with the pharmacovigilance, we are usually having the hypothesis generation. So in the hypothesis generation, we are generating that, yes, now the PVPI, usually after all the ADR monitoring centers send the reports of the individual cases to the PVPI. The PVPI would see a pattern of the ADRs across the country. And it would maybe send out a letter or a notice saying that so-and-so drug has a so-and-so reaction, which should be monitored. So that drug would be getting extra attention because there are more number of reports on that drug. So they would be sending out a letter saying that this drug should be monitored for so-and-so reaction. And from the country, it goes to the Uppsala Monitoring Center, which is the worldwide ADR reporting unit. Then comes your pharmacoepidemiology. So what happens? Do we just take the reports from the patient that you developed an ADR? Because that is the basic thing that all the 5th PharmDs and 6th PharmDs in their hospital rotations would do. One of their activities is monitoring the patients for ADRs. So is that ADR occurred? And we're reporting it. So what does it happen? Does it stop there itself? No. Logical studies, where this is going to study the interaction between the drugs and the human population. So in a real time condition. Okay, it's a real condition where the risk and benefits and use of the drugs. So here we are so pharmaceutical companies are the sponsors who spend money and they introduce a drug into the market. So they do not just leave the drug after marketing that it is not my business anymore, but they would be conducting the pharmacovigilance and pharmacoepidemiological studies to understand the risk and benefit of the drug. So now, as we can see, in a hypothesis generation, we have spontaneous reporting, intensified reporting, targeted reporting, cohort event monitoring, and EHR mining. EHR is the electronic health record. So it is a pool of, patient data. So here our suspicion is more because it's a single patient report and it reduces as we go into a pool of patients. So here, as you can see, the information is more because more number of patients, and here the information is less as it's a single patient. So now why pharmacoepidemiology? So here the researchers in the field of pharmacovigilance usually use these studies to identify the unexpected and rare safety issues. So some of the drug issues may not be seen during the clinical trials. So we are trying to identify those in the pharmacoepidemiological studies, and we are able to detect any changes in the expected ADR. So I know a drug could cause an ADR, but any changes in the frequency, that's the incidence rate increases, we want to understand with the pharmacoepidemiological studies. So the ADRs and the AEFIs. AEFIs are the adverse event following immunization. So here making it possible for the continuous monitoring of the benefit risk ratio of a drug. It can be a drug, vaccine, even the complementary or alternative medicine products also in the real world setting. So what happens with these new evidence? New evidence serves, it's important to the pharmaceutical industries to submit amendments for their approved indications for their products. So any amendment in the drug product will be done. Like maybe there's a change in the label, or maybe there is a, dear healthcare professional letter sent out, or maybe there is change in the, they may recall the drug. So such kind of amendments are important when you do the pharmacoepidemiological studies. Then even it so in your real world, you're not having a control setting as in your clinical trials. And also these study results are important to the reimbursement agencies, that is your insurance companies use this to assess of the health. Use these study results. Am I audible?
Yes, ma'am, but sometimes breaking voice is interrupting.
Okay, so yeah, that's what my internet is a little bit problematic. In case there's any, you're not able to this thing, please
Yes,
alert
yes,
me. Yes,
yes,
sir.
ma'am.
Thank you. So now we are coming to the hypothesis confirmation. So here we have the cross-sectional studies, case control studies, cohort studies, your randomized control trials, systematic reviews, and meta-analysis. So you see the bias decreases as we go towards systematic review, and the quality of evidence increases, which decreases, which is less in your cross-sectional study. So these study designs are important again in the fifth PharmD. When we have a project, we have the study design, we have to decide and we have to do. So understanding the study designs are important. So what are the pharmacoepidemiological methods that we are going to see today? So here we have our study designs. And in this study design, usually when we want to select a study design for our study, we have to have 3 questions to be asked. The first question is, what is the aim of your study? So whether you are going to give some intervention or not an intervention. So it's a descriptive means it is your patient, that's your population, and an outcome. And analytical means you have the PICO, that is the population, intervention, comparator, and outcome, or population, exposure, comparator, and outcome. So under descriptive, we have qualitative and we have cross-sectional or surveys. Qualitative can be case reports or case series. So now comes the next question that we have to ask, are we using an intervention or not in our study? So if there are 2 types again under the analytical, that is experimental and observational. So if you're going to use an intervention, then it is an experimental study where you're doing your RCTs. So I am giving some intervention to the patients. Observational, we are not doing any intervention, we are only observing the patients. So now it comes the third question. When are you observing the exposure? So now you are having first the cohort studies. Cohort studies is the after the exposure. So now exposure has happened. I'm looking for the outcome. Then comes your case control where the outcome has happened. I'm going behind retrospectively to see for the exposure. And then I have cross-sectional, which goes with the outcome and exposure at the same time. Then comes the other studies, which can be your medication utilization, evaluation studies, that is the DUE. Then safety studies, that can be spontaneous reporting, cohort event monitoring. then can be a medication adherence study. But for today, we are going to only discuss the descriptive and analytical studies. So coming to the descriptive studies, we see the case reports. So case reports are observed in a single patient, and it's usually an adverse outcome after a drug exposure to a drug. And it's usually an unusual or a novel occurrence, and it's usually important for scientific observations that are missed or undetectable in clinical trials. So like we had reported a dextromethorphan-induced dystonia. So it was just a cough syrup that caused a dystonia in an elderly patient. So this was not a known case in the drugs, monograph or their adverse event reports. So now when we come to a case series, it's a collection of individual case reports over a period of short time. So case series can be either clinical or a population based. So clinical means in a single hospital setup. So like I have seen, maybe paracetamol was taken and the patients have developed some reaction to it. So I saw the same pattern maybe in 3 or 4 patients within my practice site. So I have reported them as a case series because they have similar reaction. Population case series means it's a defined geographical area. So in a geographical area, the people are reacting in a similar fashion. So here, like an example is, they have reported a metabolic acidosis with metformin and empagliflozin, where they have shown 3 patients to suffer. So this is a case series. So advantages of this kind of a study design is, hypothesis generation, and it is informative for rare diseases with few established risk factors and usually of short duration. Disadvantages, however, cannot study cause and effect relationships and cannot assess disease frequency. Now coming to the survey, here it is called also an epidemiologic study or a prevalence study. It describes the data collected on a group of subjects at one point of time. It's a snapshot. Both the exposure and outcome variables are evaluated at the same time. So the most important question answered is what is happening right now. So coming here, we have series of different years and patients of different age group. So as you can see, different age group, but at a single year, it's a cross-sectional data. And patients of a similar, characteristics, they are all the same characteristics, they are called cohort studies. And these are different characteristics, but of the same age. They are just successive years of 30 years old, how they are, having the outcome. So we here are seeing for the cross-sectional study at a single point of time. So like this, they have used in this study to document the knowledge, attitude, practice regarding pharmacovigilance amongst the healthcare professionals. So it is a cross-sectional study. They are not going to follow them. They are just going to pick the data at a single point of time. So advantages of that, it is an easier study design because it does not have any follow-up required. It gives a good picture of the healthcare needs at a particular point of time. And it investigates multiple exposures, multiple outcomes, and it's ethically safe because you're not, intervening, like in the sense you're not giving them some new drug or something. So it is ethically safe. Disadvantages, however, it establishes association, but not causality. And it is not useful to investigate rare diseases or diseases of short duration or rare exposures. And there is a recall bias, and it's difficult to establish the time sequence of events. Confounders may be unequally distributed and group sizes may be unequal. So here confounders means, now maybe I'm doing a survey on coffee drinkers and maybe, heart disease is the outcome. So I want to see how many heart disease patients are going to have coffee consumption. But maybe I missed out the point that even that coffee drinker may be a smoker, maybe alcoholic, or maybe genetic something. So these, other things which I do not, maybe capture, but has an influence on my outcome and my exposure are called confounders. So now coming to the analytical studies, we'll first cover the observational studies. In the observational studies, there must be a comparison group. No control, there's no conclusion. Now, this is important. When we are starting a study, this is the starting point. We want to understand whether when we start a case control study, it is usually retrospective. It goes to the past. So you are having your cases and controls. Cases means they have the disease condition that you are studying. So the disease condition, the patients without disease condition, you'll go retrospectively to compare the risk factors for both the groups. In the cohort study, we have retrospective and prospective. In retrospective, we have a cohort with the disease condition. We follow them in retrospectively to see the risk factor present or absent. Prospectively, we have the risk factors, but outcome, we are following them in the future, that is prospectively, to understand the outcome. Clinical trials are also usually prospective, where they are treated, there's no treated, then they see the disease incidence, because usually they will say placebo group, but in case there is a disease condition, you can't give a placebo to a patient, it is unethical. So you would be treating them with the already existing drug therapy that the patient should be taking. So a new drug therapy, the cohort refers to individuals that share common characteristics. Here, these individuals are followed over a period of time and look for the differences of their outcomes. So usually it is the exposure groups compared to the non-exposure group. So as you can see, this diagram would explain properly that there's a start point, there's a study design, there's a study population where there's free of disease, but you are checking their risk factors present, risk factors absent, you're dividing them. Then you're following them over the period of time and seeing who has developed the disease and no disease over the period of time. So now in a cohort study, we calculate something called relative risk. So relative risk is the incidence proportion in the exposure group divided by the incidence proportion in the non-exposure group. So what is an incidence proportion? Incidence proportion is the measure of risk of disease or the probability of developing a disease over a period of time. So this is those who develop the disease, that is your outcome, divided by the entire population. So that is what we are having A divided by A plus B, that is your outcome plus the whole population. So in the exposed group and the non-exposed group. So now we have, this is our relative risk. And we will say that those who received empagliflozin had 3 times more risk to develop the outcome. So usually when we get this number, we usually will do 1 minus this number. So it comes to 2.9, which is close to 3. So we say it is 3 times more risk. Okay, so we will be understanding how to understand a risk in this, using your confidence interval. So then we also have the hazards ratio, where we are having the incidence rate in exposure group divided by the incidence rate in non-exposure group. So here we are having incidence rate, not incidence proportion. So here you can see the table is different, where we have the outcome, but we have something called person years at risk. So now what is the incidence rate? It is also called as person time rate. So it is a measure of incidence that incorporates time directly into the denominator. A person time rate is generally calculated from the long-term cohort follow-up, wherein the study participants have followed up for over a period of time, and the occurrence of new cases of disease is documented. So here each patient is followed up for until 4 of the endpoints are met, until any one of the 4. One, there is the disease has happened, so onset that the outcome has happened, or the patient left the study, or there is an end of the study. So usually incidence rate is calculated as outcomes onset divided by the person times. That is the sum of each patient, each person that was followed. So when we come here to explain what is the person time, now we have a time from 0 to 10 years. Now we will see the first person was followed from 0 to 2 years and had the outcome. Another person's starting point was 2 and was followed till 7. That makes it 5 years for this patient, and the outcome was seen within 5 years. There was another person who was followed up from here. His starting point or her starting point was 5 years from here. So it is 0, and it is like around 3 years or 4 years of time that there was an outcome. Another patient from time 0 to 10 years was followed up, but there was no outcome seen. Another patient was followed up from 2, but that is 0 for this person. So it is till 10, no outcome, 8 years. So what we would do, we will see that 3 onsets we have seen, and we are going to add for each of the patient. So 2 years plus 5 years plus 3 years plus 10 years plus 8 years. That makes us 28 years was the duration, that is person time. With 3, when we divide it something, 0.110 per person years, which is 10 per 100 person years. So we are calculating this odd hazards ratio and also seeing that they are going to get the 3 times more risk. So when you see an example, we are seeing that here an adverse drug reaction related hospitalization, a population-based cohort study. So here in a cohort means they are going to take all the patients in the hospital and see that, divide them into the patients that had an adverse event related hospitalization and those who did not have adverse event hospitalization. They would segregate the data, and then they would go back and see which were the drugs that have caused the adverse drug reactions. So this is a retrospective cohort study. So advantages is ethically safe, subjects can be matched, can establish timing and directionality of the events, eligibility criteria and events can be standardized, administratively easier and cheaper than RCTs. However, the disadvantages are also here. Exposures linked with an hidden confounder. Blinding is difficult. Randomization is not present. For rare diseases, large sample size and long follow-ups are necessary. So now coming to case controls, as the name says, we are comparing cases with the disease to a control without disease. So to identify the factors that may contribute to the medical condition. So coming to this image, you can see study starts here. We are going retrospectively, where there's a study population, case, controls. Cases, we are following them to see the risk factor present, absent, and the controls for the risk factor present, absent. So in a case control study, we are calculating something called odds ratio. So odds ratio is the odds of outcome in the exposure group divided by the odds of outcome in the non-exposure group. So here we see that it is 1.12. So when we minus 1 minus 1.12, we get something like 0.12. So it is less than 1. So there is no association identified. And even if we do not minus the 1, by seeing the confidence interval, we can identify whether there's an association or no association. So that one, this is the thing that we should see, which is important, where we will have a no effect line, that is 1, less than 1, more than 1. So we have to see whether our ratios are more equal to 1. If it is equal to 1, there is no statistical significant difference between the treatment and the control groups. So something like this, where the confidence interval is less than 1 and like the lower limit is less than 1 and the upper limit is more than 1, then it is not significant. Like you see, in this case, we have a lower limit less than 1 and an upper limit less than 1 and more than 1. So there is no association. But if there is less than 1, this says like both your upper and lower limits are less than 1, then it's significantly more likely in the treatment group than the control group. So that then we will see for the risk, the lower and upper limits to be more than 1. So if it is more than one, then significantly, the events are significantly less likely in the treatment than the control group. And now coming to an example, there's an anticholinergic drug and risk of dementia, a case control study. So as we can see that our cases are patients with dementia and patients without dementia. And we will see our risk factors retrospectively to see if they are having anticholinergic drugs before the diagnosis of dementia. So you're going retrospectively to check for the availability of anticholinergic drugs and to say that these drugs were the positive agents for the dementia. So advantage is that it is quick and cheap. It's flexible for rare diseases or those diseases which have a long lag between the exposure and outcome, and multiple possible causes of a single disease can be studied. So a rare disease which needs a long time for the outcome to be seen, a cohort study would be too long to follow a patient. So we would go with a case control study so that we already have the outcome. We go retrospectively to check for the exposures. Your disadvantage is that you rely more on the recalls and records to determine the exposure status. Again, confounding factors are here, and the selection of control group is difficult. So you have to see that if you're getting the case groups, you're able to even get the control groups. And then potential bias of recall and selection bias, that is selecting your control groups. So now coming to your analytical studies is the experimental study. So in the experimental study is your RCT, where you have your study starting, you have your study population, you randomize them into an intervention arm and a control arm. Then you have their outcome and no outcome and no outcome in both the groups are seen over the period of time that you follow them in the future. So now like we see here, there's a randomized control trial, double-blinded, placebo and placebo-controlled trial that was done. So here, when we see, usually we would write it in a PICO format. So PICO is patients, intervention, control, outcome. So in this study, they have said that the patients are going to be your adult patients with type 2 diabetes mellitus and established cardiovascular disease and other risk factors. So they want patients with these drugs, these diseases, and they are going to intervene and they're going to control. The controls are going to get placebo and the intervention arm is going to get the new drug. And they're going to follow both these groups to see the outcome, that is cardiovascular death, non-fatal myocardial infarction, non-fatal stroke, coronary revascularization, or unstable angina. Any one of these outcomes is an endpoint of the study for that patient because it's the primary outcome. So if a single patient achieves any one of this, they are terminated from the trial and they are recorded. Okay. So now advantage is that unbiased distribution of confounders, and blinding is more likely. Randomization facilitates statistical analysis. So in the clinical trials, they have randomization done by the system. So it would not be based on the person that has to, maybe the principal investigator sits and says, okay, today whoever comes will go in the intervention arm and tomorrow whoever comes will go into the control arm. It's not they who decide. So they have the softwares. So the softwares would unbiased. They will be randomizing the patients, and blinding would be there with the, who's into which arm. So there is no, bias that, yes, I want the treatment arm to have a better this one. So only the unblinded pharmacist would know which arm does a patient fall into, because that only the unblinded pharmacist has to dispense the drug. So even the vials will not be disclosed of which vial is what. So they will be coded on, only the unblinded pharmacist would know which code belongs to which group. So no one else. So the blinding is not broken. Then disadvantage is that it's expensive and it's volunteer based. So in clinical trials, all the study participants are involved, they are in volunteer based. You can't force anyone into any study. And ethically problematic at times, because in randomized control trial, you're introducing some new drug into a patient. So because of that, you don't know how the patient may react. So it is ethically more problematic than the other study designs. Then you see your level of evidence. We see that your systematic reviews are at the top. It's the highest evidence-based evidence level of evidence. And your case control and your case series and these things and all come in the bottom of the pyramid. So these things build the structure for these studies to be happening. So a systematic review attempts to collate all empirical evidence that fits a pre-specified eligibility criteria in order to answer a specific research question. So it uses explicit systematic methods that are selected with a view to minimize bias, thus providing more reliable findings from which conclusions can be drawn and decisions can be made. So a systematic review is going to take many studies put together to answer a research question. So based on the systematic reviews, research questions answered, further studies of RCTs or other pharmacoepidemiological studies can be carried out. So now we have something called meta-analysis that is part of a systematic review. So meta-analysis is the use of statistical methods to summarize the results of independent studies. So this is an example, like they are trying to understand the prevalence of adverse drug reactions in the primary care settings, a systematic review and meta-analysis. So they want to understand the prevalence of adverse drug reactions. So they will collect all the studies that have been reported from primary care settings, and they would write a systematic review. So something called a forest plot is written in a meta-analysis. So this actually gives you a bird eye view of what you want to understand from this study paper. Even if you don't want to read through the whole systematic review or the results and the discussion, just seeing this one chart in a meta-analysis can give you a brief idea about the results of the paper. So here we are having like a drug called pioglitazone, which is used in diabetic patients, but we want to see if pioglitazone has an association with bladder cancer risk in these diabetic patients. So this is going to, you have studies, your study ID. So what all studies that they have included into the meta-analysis. So these are the parts of a forest plot. Then you have your hazards ratio with the confidence interval at this end, and towards your right or right end, extreme right, is your percentage weight. Percentage weight means each study is going to contribute to this, forest plot. So how much is the weight of a study is shown in this, section. So what does this, how would you read it? That is, you will first see your line 1, that is your no effect line. Then you have some horizontal lines. These horizontal lines are your study, each study. Now you see each line is not of the same size. Now you can see if you have a sample, this square box which is seen are your sample size of your study. Okay, so bigger the sample size, bigger is the square. Okay, you see square is big, this number is more. So in this case, if you see 16 and 1, this is the highest weight and this is the lowest weight. So you see this one, it has a big square, and this one you can barely see the square. So square, bigger the square, bigger is the sample size. Then we have something this called like a diamond shape that's at the bottom. This diamond is the average weight, that is the total weight of all the studies. So you see this diamonds, this end and this end represent the confidence interval. Okay. So now when you see the weight, larger the weight, more is the sample size, narrow is the confidence interval. Okay. So larger the size, weight, smaller, larger the sample size, narrow the, confidence interval. See, smaller the weight, smaller the sample size, wider is the confidence interval. So you can see this one's confidence interval is so long. Now, if you see, this one is below 1 and more than 1. This again is below 1, more than 1. below 1, more than 1, but this has only both above 1. This again is below 1, more than 1, and again this is below 1, more than 1. But here this again is all at above 1, and this is again below 1 and more than 1. So in this overall, this one, it sees that only 2 of the studies fall more than 1. So even you see, it says favors non-pioglitazone, favors pioglitazone. So now it says that this hypothesis of it causing cancer can be ruled out because our analysis does not give you, significant values. So now you can safely say that there is no risk. So maybe you can conduct any other further data, study designs to understand if, yes, this may cause, like these are already published studies. You can conduct a newer study to see if there are any new outcomes. Then now that much is for the signals that you're going to confirm, how you're going to confirm your signals. So coming to the career options, if you see in pharmacovigilance alone, you have like a clinical research scientist. So clinical research scientist means they're having now a clinical research happening. So actually pharmacovigilance starts way from the clinical research point itself. So when we are having a clinical trial happening, we do monitor patients for adverse outcomes because it's a novel drug. So a new drug. So when we are giving them, this clinical research scientist would be assessing the safety during the clinical trials, would help in monitoring and assessing the safety profiles of the investigational drug. So you have a role in the clinical trials itself, not just in the post-market. Then comes your drug safety associate or pharmacovigilance associate, who is responsible for collecting, reviewing, evaluating safety data related to the medications or medical products. Now, even not just pharmacovigilance, even the material-vigilance, that is your medical devices, are, being a new stream of this vigilance is coming up. That the medical devices related adverse events are also reported. So that is also part of pharmacovigilance, which will also be, your drug safety associate can be doing. Then your drug safety scientists and pharmacovigilance scientists, these do more in-depth safety data analysis. So here what they're going to do, they're going to assess the risk assessment, preparing regulatory reports, and they collaborate with a multidisciplinary team, and they ensure that the safety profile of the drug products are taken care of. Then you have a medical reviewer. A medical reviewer is usually who's going to review all the, you send your ADRs. So their ADRs, they're going to assess these adverse event reports, the medical literature, that is like, these case series, case reports, they are monitored by the, companies. They will be sending out to the corresponding author mails, asking them for further information on what drug company was it, because in your case reports, you're not going to mention your drug brand name. You're just going to mention the generic name. So the drug, if there is n number of companies, so all the company, medical reviewer will send out a mail asking which drug company was it, what was the batch number, what was the expiry date. So such kind of extra informations would be collected. So that is the, a constant review of the literature would be happening. And they would be understanding the potential causal relationship between the reported events and the medications. Then something consultant or happens when you are, you have worked and you have an experience in the field of pharmacovigilance over the period of time. So you can, start a consultancy company for yourself where you can provide these services for companies. Then you have pharmaceutical industry liaison, where you are going to liaise between the pharmaceutical companies and the regulatory authorities, so that you're going to ensure that the safety data is accurately communicated to the regulatory agencies and facilitate interaction during safety assessment. Then comes your pharmaceutical risk management specialist. So your risk management specialist, as it says, risk management. So you're going to focus on designing and implementing risk management plans. So for the pharmaceutical products, they work to mitigate potential risks and ensure ongoing safety of medications in the market. Then comes your pharmacovigilance auditor. So now auditor, as the term says, you're going to audit. You'll audit the company to understand if their systems are in compliance with the regulatory standards. And you can, assess the areas. You'll give a preventive actions and corrective actions, such kind of things. Then you will have your pharmacovigilance manager or a team lead, where you're going to, lead a team of, professionals who are responsible for the pharmacovigilance activity in a company. And you're going to, coordinate, collect, and analyze the safety data, ensuring compliances with the regulations. Then you have your regulation affairs specialist, where you're going to, ensure that the pharmaceutical product meets the regulatory requirement, and, has collaborating with the pharmacovigilance team and providing the relevant safety information to the regulatory authorities. Actually, these are few just to mention. There are a lot of other even opportunities available in the market, like, you have your CDMs, and like as Sir said before, being a PharmD grad, even you can get into research if you are interested. So you can take a PhD program, or else you can, even if you're interested in academics, you can go for your teaching, or else some students do MS programs in, maybe biotechnology, maybe regulatory affairs, it can be biopharmaceutics. So there are a lot of new openings and, venues for a PharmD grad to explore. And I think we shouldn't, narrow down our opportunities, saying that in India, we do not have, scope for PharmD grads. We have, it's just, we have to, find the right way to get into the things. And I think we should first get the, knowledge about it, and then we can, really get into the right job, and it would be a really wonderful thing. And even as a CRC, you can serve as a CRC or a CRA, which is a clinical research coordinator or a clinical research associate. So a CRC would be confined to the study site. So you will be a connecting point between the CRO, that is your clinical research organization, and your principal investigator, because from the site initiation till the site closeout, you're going to take care of all the activities, whether it is monitoring, it is a team where you are going to take care of everything. That is, the process is going in a correct direction, is the protocol followed or not. And a CRA is a person above the CRC, because a CRC reports to a CRA. So a CRA is a clinical research associate. So he is going to come to the site and he's going to check if everything is happening according to the protocol. So that's something called site monitorings will be done. So site monitorings, that one clinical research associate will be having n number of sites under him, but a CRC will have only one site. So even clinical research is a good area for you to explore, because even CDMs, that is clinical data management, data analysts, data scientists are also the upcoming, job opportunities for the PharmD graduates. So I think I'm done with my talk. Thank you so much. If any questions, I'm open for it.
Yes, students, now it is your time to clarify your doubts, questions. From 4th B Pharmacy, 5th PharmD, 6th PharmD. Students, be active and clarify your doubts. Fifth year PharmD students. Any question regarding the pharmacoepidemiological research? Till now, the ma'am explained very detailed about the various pharmacoepidemiological studies, how to confirm the signals of drug reactions. And this year you have project works also you are planning, and already some people have started some works. Any question? Even you can drop your question in a chat box also. 6th years, those are on online basis. 6th year entrance, you can drop your questions, anything in a chat box also. Fourth B Pharmacy and fifth Pharmacy students, is there any questions to discuss? Okay then. Thank you, ma'am, for your elaborated and valuable, informative, detailed lecture on pharmacovigilance research methods. Of course, the opportunities in pharmacovigilance. It is very useful for our students for understanding what is happening in pharmacovigilance and what roles they have in future if they want to choose pharmacovigilance as a career. And the basics of pharmacoepidemiological studies, they understand. It's a really good lecture. Thank you for your valuable time and accepting our request and spending time to give a valuable lecture for us. Thank you very much, ma'am.
Thank you so much, sir. Thank you for the opportunity as well. Thank you.
Thank you. With this, we are closing this guest lecture section. Thank you, one and all. Madam, లేదు మీరు షట్ డౌన్ చేసి సిస్టమ్ కూడా అన్ని కనెక్షన్స్ అన్ని రిమూవ్ చేసి షట్ డౌన్ చేసి అక్కడ బ్యాగ్ ఉంటుంది చూడండి, బ్యాగ్ లో పెట్టేసేయండి. అండ్ ఎవరన్నా స్టూడెంట్ దగ్గర ఆఫీస్కి సెండ్ చేసేయండి, కుస్మాకు వచ్చామని చెప్పేసి. ఎవరన్నా ఒక స్టూడెంట్ దగ్గర కుస్మాక్ మేడం సిస్టమ్ షట్ డౌన్ చేసి బ్యాగ్ ఉంటుంది చూడండి, ఆడ బ్యాగ్ లో పెట్టేసి స్టూడెంట్ దగ్గర సెండ్ చేసేయండి, కుస్మాకి ఇచ్చామని ఆఫీస్లో సబ్మిట్ చేయమని. ఉంది చూడండి అక్కడ టేబుల్ పైన ఛార్జర్ వైర్ కూడా ఉంది కదా, ఆ ప్లగ్ పాయింట్ లో పెట్టున్నాం కదా ఛార్జింగ్ వైర్ కూడా. హలో.